Hello everyone, in our previous video we learned about how to generate taxa bar plots in QIIME 2. In today's video we are going to do alpha diversity analysis after this taxa bar plots generation. So without any further delay, let's start today's video. Once you have got your taxa bar plots in QIIME 2, the next step towards performing alpha diversity analysis is getting a rooted-tree.qza file. So this rooted-tree.qza file is basically a phylogenetic tree which you get in QIIME 2. So this is very critical because this will provide you various quantitative metrics for performing a statistical analysis in QIIME 2. So you can simply type the command for getting this rooted-tree.qza file. You can see this on your screen. Once you have typed this, just press enter and the system is going to take some time to process this data. So the time taken for processing this request or processing this command will depend upon the RAM and the ROM availability. So as you can see, we have got all these required files. So in the next step we are going to use this rooted-tree.qza file in order to perform alpha diversity analysis. Once you have got this rooted-tree.qza file, the next step is to perform alpha diversity. You can get the alpha diversity matrix individually as well as collectively. So I am going to provide you a single command by which you can get all the quantitative measures related to alpha diversity in a single framework. So for this, first type the command QIIME space diversity space core dash matrix dash phylogenetic space backslash enter. The next line type I dash table, input your table, that is table.qza, which is the correct name of your file. The next line type dash dash I dash phylogeny and then type rooted dash tree.qza space backslash, press enter and then you need to type your sampling depth command for sampling depth dash dash p dash sampling dash depth depth. I choose the value 11,000 based upon my sample as I have discussed earlier, then press enter. In the next line you need to provide your metadata file dash dash m dash metadata dash file. Input your metadata file name correctly and then again space backslash enter and then you need to type your output and the output will be output directory. So dash dash output dash dir space core dash matrix dash results results. One thing which is very important for you is that you can get all the metrics or all the parameters related to alpha diversity individually or collectively. The command which I am showing you here provides all the parameters related to alpha diversity in a single format or in a single framework. So if you need to know about individual commands, I would provide all that in a separate file. The link for that is provided in the description and your data will be processed very shortly. As I'm saying, you will get all the parameters or all the metrics related to alpha and beta diversity, like you get rear file table.qza faith underscore pd underscore vector observed features Shannon vector, evenness vector, unifrac distance. So many of these relate to a beta diversity. I will discuss about beta diversity in another video. So for now, we are going to use the alpha diversity parameters, that is observed features, Shannon index and Simpson index. One more thing which I would like to discuss here is that this command does not yield the Simpson index. For that you need to provide a separate command, as I'm going to show you later in this video. So once you have you have got your alpha diversity parameters, it is first important to validate your results, whether the data you have got, whether it is correct or not. So to validate your alpha diversity matrix, you need to first check this using a rarefaction command as shown on your screen, that is QIIME space diversity space alpha dash rarefaction space backslash. To continue with the command, you need to first input your table, that is table.qza space backslash enter, then dash dash I dash Phylogenetic rooted dash tree.qza space backslash press enter, then dash dash p dash maximum depth 2000 space backslash press enter, then dash dash m dash metadata dash file, and you need to input your metadata file name very correctly, and then finally you need to basically provide the output command for visualizing your alpha rarefaction, and ultimately this command is going to provide you a QZV file, that is alpha underscore rarefaction dot QZV, and we are going to analyze this QZV file in the view.qime2.org. Now we have got the alpha and underscore rarefaction dot QZV. Let's move to our browser window and analyze this file in the view.qime2.org browser. You select the file and open your this alpha underscore rarefaction.qzb file. You will see that your observed underscore features metric is showing a plateau behavior around your sampling depth value. You can see here in this curve, observed underscore feature is basically placed curve has been drawn for alpha underscore feature versus the sequencing depth or sampling depth. So it around your sampling depth of 11,000, it is showing a plateau behavior, which means that your analyzed data is perfectly correct, and we are now ready to move to the next step. So now, since we have verified our alpha diversity data, it's now time to visualize all the metrics related to alpha diversity. So in this first step, we are going to analyze Shannon underscore vector dot QZA file and will convert it into .qzv file. Once we get this .qzv file, we are going to analyze this into the view.qime2.org browser window. So to convert this Shannon underscore vector dot QZA into .qzv, type the command QIIME space diversity space alpha dash group dash significance space backslash press enter. In the next line, type the command dash dash I dash alpha dash diversity core dash matrix dash results. You got this folder earlier. Basically you need to input your file name properly where it is located and then press enter. In the next line you need to input your metadata file name properly, and finally you need to visualize your data. Finally, since you are going Shannon underscore vector, so you will convert into Shannon underscore group underscore significance dot QZV, press enter, and you got this Shannon underscore group underscore significance dot QZV file. So likewise, we are going to type the command for all other alpha diversity matrix such as observed features and PD vectors and other items. So next type QIIME space diversity space alpha dash group dash significance space backslash press enter, then type the command dash dash I dash alpha dash diversity faith underscore PD underscore vector underscore QZA. Again type, input your metadata file name properly, ensure that the spacings and the formattings are proper, and finally visualize your data properly. Likewise, you can also input the command for your observed underscore features as shown on your screen. QIIME, you will type the command QIIME space diversity alpha dash group dash significance, then again input your file name properly. What is the file name you got? That is observed underscore features underscore vector dot QZA space backslash press enter. Again you need to input your metadata file, then provide the output command for visualizing your data. Just convert it into a QZV file here and just press enter. Fine. So you've got three parameters. One parameter is pending, that is Simpson index. For that you need to provide a separate command. For getting this Simpson index, you need to first rarefy your feature table, and for this purpose type the command QIIME space feature dash table space rarefy rarefy space backslash press enter. In the next line, type the command that is for input your table, that is ii dash dash I dash table table dot qza In the next line, provide your sampling depth, that is dash dash p dash sampling dash depth depth 11000 space backslash, press enter and provide the command for getting the output, and the output will be dash dash o means output dash rarefied rarefied table, and you get table underscore rarefied dot qza, then press enter and you got this table underscore rarefied dot qza, and we are going to use this rarefied table for getting the Simpson index. So getting this Simpson index, first type the command QIIME space diversity alpha, simply because it is an alpha diversity matrix. Then in the next line, always you have to import your table, that is table underscore rarefied dot qza space backslash. So you have to first input your table. In the next line, you have to provide your sampling, your metric, which you need to basically identify. Here in this case, this is Simpson index, and finally you need to output your metric. And for getting this output metric, you need to type the command dash dash o dash alpha dash diversity Simpson underscore vector dot qza. You will get the Simpson vector as well. Fine. Now we are going to convert this Simpson underscore vector dot qza into a qzb file. Converting this .qza into .qzb file is very simple and straightforward, as we have done for all other parameters earlier. Simply copy paste this command QIIME space diversity space alpha dash group dash significance space backslash, press enter. Again you need to input your file name, that is alpha dash diversity, and then input your file name properly, Simpson underscore vector dot qza space backslash, enter. Again you have to type your metadata file. In the next line, dash dash m dash metadata dash file, and input your metadata file name properly. And finally you need to get dash dash simply your visualization of your data, that is dash dash o dash visualization, type Simpson underscore vector dot qzv, and you can simply press enter. Here you will get all these files. Now we are going to visualize all these qzb files one on one, so that you can understand the meaning of all these parameters properly. So now let's analyze all the data or all the parameters we got in the previous step. So in the very first step here, we are going to analyze observed underscore features underscore group underscore significance dot qzb, or observed underscore features dot qzb, whatever the file label you have used. So simply open your view.qime2.org window and then select your desired file. I will select observed underscore features underscore vector dot qzb, whatever I've labeled it. Okay, fine. So once you opened this window, you are going to see the alpha diversity box plots. All these figures, whenever all these parameters rather, when you open them into the .qzb into the view.qime2.org window, they are going to show you box plots and some statistical parameters in the form of Kruskal-Wallis tests. These box plots are further analyzed using two parameters, two statistical parameters. One is capital H or H value, and second is the P value. So H value provides you information about the richness into your data. Higher the H value, higher will be the richness of the species, means you will get more number of species. Fine. So in this case, our H value is 4.266 for all the groups combined together, and the P value is 0.23407. As you might be aware that for any data to be statistically significant, the P value should be less than 0.05. Okay. However, in this case, the P value is very much higher than the suggested or the standardized value. So it means for all the groups combined, the P value is insignificant, it means that this data is not statistically valid. Moving downwards, we can also get the Kruskal-Wallis parameters in the pairwise format, and again you will get three parameters here: H value, P value and Q value. Q value is rather adjusted or corrected P value. Again, the Q value or P value should be less than 0.05. So in this case, you can see that for all the datas or all the groups, that is group 1 and group 2 here, as shown on your screen, the p value is again far higher than the suggested or standardized value of 0.05. So it means this data is statistically not significant. The reason being here is that the highest n value or highest number of species or significant sequences in our data is 5. In majority cases, the n value is 1. So that is why we are not getting the significant p value as it is desirable. So more the number of samples are there into your data and more is the number of species, the p value would be adjusted towards the standardized data. So I hope you have understood this meaning of observed underscore features, and in the next step we are going to analyze the Shannon underscore vector dot vector underscore group underscore significance dot QZB. Now again, click on this, drag and drop or click here once you have opened view.qime2.org, and go to your desired folder and select Shannon underscore group underscore significance dot QZB. Like the observed underscore features underscore group underscore significance dot QZB, you are again going to the alpha diversity box plot for this particular metric, and moving downward you will get the Kruskal-Wallis parameters, that is H and P value. I've told you earlier, the higher the H value, more is the species richness or sampling richness, or rather sequence richness in case of amplicon sequences. Higher the H value, higher is the species richness. But again, P value is not significant. Moving downward, the Kruskal-Wallis parameters for group wise or rather pair wise, that is in group 1 and group 2, if you are comparing them, the H value is again like increasing from 0 to and up, achieving a maximum value of 2.14, whereas the P value, it is still not significant as the values are far higher. In fact, for Q value as well, the both the P values and Q values are statistically insignificant because they are far more exceeding the suggested values of the standardized value of 0.05. So I hope you have understood the meaning of all these parameters, although I have explained all these things in detail earlier as well. So in case you have still any doubt, you can please let me know in the comment box, and if you do end up like our content, then please don't forget to hit the bell icon, and also please subscribe our channel. We will be eagerly waiting for your comments and suggestions so that we can improve our content for all future videos. Thank you so much again for watching our video.